Slide görüntü mü?
Yeah.
The second, genotype and the phenotype of microorganisms. The material basis of heredity, which determines the genetic properties of microorganisms, is DNA, desoxyribonucleic acid, a fragment of DNA molecule that controls the synthesis of a single protein is called a gene. The genes are encoded with the genetic information about all the properties of the cell, the shape, structure of proteins, and their functions, and is a complete set of genes that the cell processes represents its genotype. The genotype determines the potential manifestation of the properties of the cell of microorganisms. And the next slide. The properties of microorganisms that are shown in certain conditions of their existence are called a phenotype. In other words, the phenotype is the sum of traits defined by the genotype and implemented in the specific environmental conditions. And depending on the conditions, microorganisms of the same genotype can form individuals with different phenotypes. The bacterial phenotype is indicated by the same symbols of the genotype, but the first letter is capitalized: histidine, arginine, and etc. And the next third stage is the forms of the microbial variability. Variability of microorganisms is divided into heredity due to genotype change and non-heredity. It is a phenotype. Phenotype change is a variability microbes formed from a single mother cell may differ in enzymatic activity, morphological characteristics, and the need for food source. And the phenotype variability includes the adaptation, desiccation, modification. Adaptation is a adaptation of microorganisms to new environmental conditions. At present, this phenomenon is not due to the change in the microbial cells, but to the development of previously modified individuals and the death of unsuitable ones. Desiccation is a cultural variability when, for example, from a pure culture found on a dense environment, colonies that differ sharply in the morphological structures. It may be a type smooth, type of rose, type of slimy, and the others. And the modification. Modification is a change of microorganisms under the influence of the environmental conditions. Only phenotype features, it may be shape, size, color of colonies, and others. Modification is observed in normal conditions of life, and it is a reaction of to the external stimuli that would not associate with the volition of physiological processes in the body, and the modification change easily differ when the conditions that cause them of eliminated. Next slide. Genotype change of microorganisms. The variability of microbial traits caused by the restriction of the genetic apertures is manifested in the form of mutations and the genetic recombination. It may be combinative change. And the mutation. Mutation are sudden, abrupt change in the gene. The process of mutating genes leads to the change as inherited and rises, and even the factors that caused some cases to uh, and the spontaneous mutation without the direct action are very rare, about in 100,000, and they are characterized by a change in any feature and usually stable. And the inductive of mutagenic mutation occur to the environmental factors, and they are relatively common. And the ultraviolet, X-ray, and the radioactive radiation have mutagenic effects, which cause damage to the genetic apparatus of the cell. And the chemical mutations include powerful substance, toxic, medical acids, and etc. In prokaryotes, combinative change is including transduction, conjugation, and the transformation, and the others. Transformation is a transfer of genetic information from the donor bacterium to the recipient cell. The most effective transformation occur in bacteria of the same species or closely related species, and in this case, the recipient's chromosome includes only one strand of the donor's DNA molecules with the formation of the molecular heterozygote. And usually a bacterial cell acquires on the property as a result of transformation, and with the help to the transformation, DNA molecules are based on the criteria such as capsularity, enzymatic activity, resistance to the poison, antibiotics, and etc. Transduction, the transfer of genes from a bacterial donor cell to the recipient cell by means of the modulate phage, and during transduction is possible to transfer gene that control the nutrition of bacteria and the other subpartites. And conjugation is a form of the sexual process in which male and female microbial cells connected and exchange nuclear matter between the through a cytoplasmatic right from between the cells. And in this case, the genetic material of the donor cell passes into the recipient cell. After recombinant and the cell division forms with the signs of conjugation, cells are formed. As a result of genetic recombinant, new bacterial recombinant cells are formed which have the inherited characteristics of both parents. And the fourth stage is a practical significance of microbiology variability. Heredity and variability is inseparable linked categories of biological phenomena that determine the evolutionary development of living organisms at any level of biological organization. As a result, the doctrine of heredity and the variability of microorganisms is a scientific basis for the systematics of microorganisms and their identification, and the knowledge of the regulators of modification and mutation variability makes it possible to carry out purposeful selection from a population of microorganisms of individuals with the necessary properties for person. In this way, highly active strains of many producers of various organic compounds were obtained. And next slide, selection of microorganisms. For the selection of useful mutation is carried out in several ways. And they are through the search and the selection of useful forms of microorganisms from natural sources, and as a result of adaptation of microorganisms by growing under constantly changing cultivation conditions, and through repeated isolation of pure culture from the production strains by selection induced strains, by using the phenomena of transformation, transduction, and the conjugation to the better strains with a new properties. And currently, a new direction of molecular biology has been developed with genetic engineering. And the genetic engineering deals with the construction, installation, and the transfer of certain genes from one cell to another. As a result, cells acquire new properties. And in this slide, you can see the list of reference to explore, and all materials and all books in the our library sites, and you can use this for your lecture. And thank you for attention. If you have questions, please ask. Сұрақтарыңыз, вопросы есть?
А вы скинете презентацию?
Нету.
Да, я вам сейчас отправлю на WhatsApp. Потом насчет
Хорошо.
сказать вам. Вы первый СРСП уже отправили, да? Только не все студенты.
Да.
Не все. Я открыла доступ на второй СРСП. Слышите меня?
Да, слышим.
За первый СРСП я поставлю сегодня баллы. У кого, если нет баллов, то они уже не могут эти баллы уже взять.
А кто уже скинул и первый, и второй?